Welcome to Science Era. In this video, we're going to discuss phagocytosis. Three things you need to understand by the end of this video is to explain the method by which pathogens are recognized by phagocytes. You should be able to describe the process of autophagy and phagocytosis, forecast how biochemical activities within the phagocyte result in pathogen destruction. I will be using Prescott's Microbiology 11th edition to explain the process of phagocytosis. Let's begin. Phagocytosis. During their lifetimes, humans and other vertebrates encounter many microbial species, but only a few of these can grow and cause disease in otherwise healthy hosts. Phagocytic cells, macrophages, dendritic cells, and neutrophils are an important early defense against invading microorganisms. These cells recognize, ingest, and kill cellular microbial species by the process called phagocytosis, and intracellular parasites by the process autophagocytosis. Phagocytic cells use two basic molecular mechanisms to recognize microorganisms. First one is opsonin independent or non-opsonic recognition, and the second one is opsonin dependent or opsonic recognition. They then encase microorganism within a killing chamber which unites with lysosome to facilitate proteolytic degradation of microbes. We are now going to discuss non-opsonic recognition of foreignness, and we're going to provide more details regarding the result of pattern recognition by phagocytes. Recognition of foreignness. The figure on the screen represents the recognition of pathogen-associated molecular pattern, also known as PAMs, by pattern recognition receptors. PAMs bind PRRs, especially toll-like receptor, mannose receptor, glucose receptors. PRR binding results in signaling the upregulates cytokines gene expression through a common NF-κB signal transduction pathway. So, what is recognition of foreigners? Opsonin independent mechanisms are receptor-based systems with germline encoded molecular pattern that activate phagocyte. The phagocyte surface is littered with membrane-bound pattern recognition receptors, also known as PRRs, that can quickly activate their ligands when the phagocyte investigates its surrounding. Other PRRs remain in the cytoplasm of the cell, examining the intracellular environment of the sign of invasion or damage, while each PRR recognizes a distinct pattern. Once attached, the PRR can assist cytokines enzymatic activation or transmit a signal to a common transcription activation pathway, upregulating cytokine expression. The multi-lectin protein is one form of PRR that binds to carbohydrate generated by infection. The mannose receptor, which is present on dendritic cells and macrophages, and identifies and binds the repeated mannose, fu- fructose, or an acetyl glucosamine component of a variety of bacteria is one example of multi-lectin PRR. Some multi-lectin PRRs bind glucan on a variety of fungi, whereas other binds mannose on Mycobacterium tuberculosis. So what happened is phagocytic surface contain PRRs, pattern recognition receptors, which activate their ligand when phagocytes investigates the surrounding. PRR which remain in cytoplasm examine the intracellular environment for invasion or damage, while each PRR recognize a distinct pattern. And PRR can also assist cytokine enzymatic activation or transmit a signal to a common transcription activation pathway, which can help you in upregulating cytokine expression. Another type of PRR is soluble and remains in cytoplasm of phagocytes, known as nucleotide binding and oligomerization domain or NOD receptors. They sense endogenous metabolites and regulate programmed cell death, or also known as apoptosis. Apoptosis. A survey of human genome predict approximately 22 similar proteins known as nod-like receptors (NLRs). NLRs also detect cytosolic pathogen-associated molecular patterns, such as terminal meso-DAP of gram-negative peptidoglycan and host molecule called damage-associated molecular pattern, also known as DAMPs, such as uric acid and heat shock proteins. DAMPs are diverse group of proteins that arise from cellular injury and stress. NLR binding to a specific PAM or DAMP result in oligomerization of several NLRs, recruiting cofactor and the enzyme capase 1. This NLR capase complex is called the inflammasome because it enhances the inflammation when capase 1 cleaves and activates pro-inflammatory cytokines, especially interleukin. Interleukin gene expression is extended by extracellular signal transduced through a multitude of membrane-bound receptors that share a similar transcription factor when interleukin is activated in response to an intracellular NLR signal. The figure on the screen represents cooperation between two pattern recognition receptors (PRRs) binding of a toll-like and nod-like receptor TLR or NLR respectively by pathogen-associated molecular patterns. PAMs activate two different response mechanisms that cooperate to produce a potent pro-inflammatory cytokine interleukin. Binding of PAMs to TLR's toll-like receptors activates an ancient signal transduction system that releases the NFKB transcription factor to upregulate gene expression of pro-IL interleukin 1. Importantly, binding of other PAMs cathepsin or reactive oxygen species to NLR result in oligomerization of several NLRs, which then binds cofactor to assemble and activate proteolytic enzyme capase, cleaves the pro-interleukin into the active interleukin cytokine, upregulating numerous inflammatory cells and biochemical pathway. So the purpose of nod-like receptor is to detect endogenous chemical and govern programmed cell death or apoptosis, and they are also capable of detecting cytosolic pathogen associated molecular patterns. Next we have is toll-like receptors. Toll-like receptors or TLRs identify and bind specific PAMs from various pathogen classes, for example viruses, bacteria and fungi. They convey this binding to a nucleus of the host cell to induce proper gene expressions and host response. TLR and multi-lectin PRR ligand interaction activates the transcription factor NFKB by degrading its inhibitor IKB in a similar evolutionary ancient signaling pathway. Later in an infection, NFKB stimulates the expression of a range of a gene, including gene of cytokines, chemokines, and co-stimulatory molecules, all of which are important in initiating and directing adaptive immune response. As a result, the binding of certain microbial components to phagocyte receptors is a crucial first step in the clearance of foreign materials and pathogens. The germs or its components are sequestered within a membrane enclosed compartment which is subsequently linked to a lysosome to facilitate microbial death and digestion. Intracellular digestion imports extracellular particles via extension of its cell membrane as pseudopodia to end up with an internal phagosome. This process initiates phagocytosis. Other processes begin when intracellular microorganisms or their products are coated with ubiquitin like that of protein recycling by proteasomes. Ubiquitin labels germs for phagophores, capture in this application free floating or open membrane. The phagophores then surrounds the microbe forming a double membrane autophagosomes. Autophagocytosis or autophagy begins with the formation of autophagosomes. The figure on the right represents how autophagy is similar to phagocytosis. Intracellular parasites such as viruses and bacteria that escape phagosome are captured by free-floating membrane that form a phagophore. Cytosolic ubiquitin molecules coat the intercellular invaders and their macromolecules. The ubiquitinated particles are then surrounded by phagophore to form an autophagosome. Like phagosomes that begin that phagolysosome during phagocytosis, autophagosomes or autosomes fuses to lysosome so as to digest its content. Once the lysosome fuses with autosome, it is called autolysosome. Both phagolysosome and autolysosome can have some of their debris donated to endoplasmic reticulum, where antigenic peptides are further processed to bind MHC molecule destined for the phagocytic or phagocytes cell membrane. The remaining debris is recycled and released to the environment. Let's look at the process of phagocytosis. Figure A represents a receptor on phagocytic cell such as macrophages and the corresponding PAMs participating in phagocytosis. The process of phagocytosis includes B. Ingestion. In C. Participation of primary and secondary granules and oxygen dependent killing event. In D intracellular digestion, and in E. exocytosis, as this autophagy digested fragment can also enter endoplasmic reticulum to be processed for antigen presentation. LPS receptor lipopolysaccharides receptor or TLRs, toll-like receptors or MHC1 class 1 major histocompatibility protein or 2 class 2 major histocompatibility protein PAMS pathogen associated molecular patterns. Exocytosis process used by neutrophil to expel microbial fragments after they have been digested is known as exocytosis. Phagolysosomes unite with cell membrane. results in extracellular release of microbial fragments. Macrophages and dendritic cells undergo process of antigen presentation which move fragments from phagolysosome to endoplasmic reticulum. Peptide fragment components combine with glycoprotein become parts of the cell membrane. Peptide bound, so they are ultimately presented outward from the cell. This brings us to the end of phagocytosis. Thank you for watching. If you like my video, don't forget to subscribe for more.